Hello guys and welcome back to Agriculture Academy. So as part of the carbohydrate metabolism, till now we were discussing about the glycolysis. Sorry. So in today's lecture, we will be talking about gluconeogenesis. So what does gluconeogenesis means? It means synthesis of glucose. Okay. So like from where the glucose will be synthesized inside the body. See, of course, you know glucose is made up of carbohydrate, hydrogen, and oxygen, right? But inside our body, we have other sources from where the glucose can be synthesized. For example, let's talk about lactate. So in the previous lecture, we saw that from glucose under glycolysis, you get pyruvate, and pyruvate gives you lactate, right? So we can use lactate also, we can use glycerol also, and amino acids. So all these can be used in order to synthesize glucose. And that's why, guys, when like you are suppose fasting, so eventually, suppose you do fasting for more than 10 days and you're not eating anything. So what will happen? All of your glycogen storage will be gone, right? Your fatty acids will be started using, right? Your adipose tissue will be used. And still, if you're not eating, then what will happen is that finally your muscles will start to break. Now, what does it mean? So muscles, we know that it is made up of mainly proteins. Now, in order to survive, this protein will be degraded to amino acids, and these amino acids will go for the synthesis of glucose. And that's how you can survive without food for about a month. Okay. So let's talk about gluconeogenesis in detail. So gluconeogenesis is a generation of glucose from certain non-carbohydrate carbon source. Okay. Non-carbohydrate. So for example, like amino acids. During the breakdown of proteins, glucogenic amino acids, although not ketogenic amino acids, form glucose. This you will understand more once we do this entire gluconeogenesis, like what do you mean by glucogenic amino acids. So glucogenic amino acids means these amino acids which go for the synthesis of glucose. Okay, and this will go for the synthesis of ketone bodies. So from the breakdown of lipids such as triacylglycerides produces glycerol and fatty acid. Glycerol is then converted to glucose. Pyruvate and lactate is also from they also form glucose. So all these substances, they must be converted to first oxaloacetate, and there is no pathway in the animal for the net conversion of acetyl-CoA to oxaloacetate. So what does it mean? Where do you see oxaloacetate? You see in the Krebs cycle, right? So we know that acetyl-CoA is there. One second, let me write it once again. Okay, so we know that glucose is there. Under glycolysis, glucose will give you pyruvate. Okay, from pyruvate you will get your lactate or acetyl-CoA. Now, acetyl-CoA you will get in the presence of oxygen, right? This is in the anaerobic condition, the lactate. Now, this acetyl-CoA will undergo TCA cycle and form oxaloacetate. But if suppose I want to go for the gluconeogenesis, like where the glucose needs to be synthesized, okay? So what will be the best way? That acetyl-CoA should go back and form the pyruvate, right? The oxaloacetate in the TCA cycle should become the acetyl-CoA, but that is not happening because we don't have any enzymes which can do that. What? The conversion of acetyl-CoA to oxaloacetate. And that's why our body, our cell needs to find the alternate sources for the glucose synthesis. Okay. Guys, is this much clear? Should I explain once again?
No, ma'am, it's clear.
Clear, right? Okay. So we know that brain uses glucose only as source of energy. Ketone bodies. Okay. So these ketone bodies are also used by the brain as energy source during the times of fasting. Testes, erythrocytes, kidney, medulla, they also depend upon the glucose as the energy source. Now this is your gluconeogenesis. I couldn't get the better image, so let's try to focus here only. Okay, so first you can see that here this is your glycolysis, and here we have got the gluconeogenesis. So we do see that it is the reverse of the glycolysis, but never mentioned in front of any teacher or any researcher or scientist, because the steps are not exactly reversed. You have got different enzymes performing the different steps. For example, the three irreversible steps which I have mentioned, those are done by the different enzymes. Rest all can be done by the same enzymes. Okay. So your first one. So we know that glucose to glucose 6-phosphate, it is the hexokinase, your first one. right? Then let's see the glycolysis first, and then we'll go with the gluconeogenesis. So glucose 6-phosphate to fructose 6-phosphate, your phosphoglucose isomerase. Then you have got the PFK, fructose 1,6-bisphosphate, right? Then aldolase to give you glyceraldehyde 3-phosphate and dihydroxyacetone. Then you get the glyceraldehyde 3-phosphate dehydrogenase will give you 1,3-bisphosphoglycerate. Then again kinase 3-phosphoglycerate, mutase 3,2-phosphoglycerate, enolase will give you phosphoenol pyruvate, and then pyruvate kinase, like we have discussed about this enzyme in detail yesterday, will give you pyruvate. So this is your glycolysis. Now let's check the gluconeogenesis. Okay, so what is happening in the gluconeogenesis? From where you're going to start? You're going to start from the bottom. So what is the first thing? Whatever source you get, okay, the pyruvate we have got. So first thing will be to convert this pyruvate back to your phosphoenol pyruvate, right? And this cannot be done directly. So your pyruvate will be converted to oxaloacetate, and then oxaloacetate will be converted to phosphoenol pyruvate. So guys, how many enzymes are required now? 2 enzymes are required, जो हमने पहले एक step में कर दिया था. Like only one enzyme was required over here, but over here 2 enzymes are required. And look at the amount of energy also required over here. So you can see that pyruvate to oxaloacetate, you see that ATP is required, okay, and CO2 is being released. It is added pyruvate to oxaloacetate. So pyruvate is your 2 carbon, right? Oxaloacetate is your, I think, 4 carbon. Okay. Then what do you see is, do you see any phosphate group attached to oxaloacetate, guys? Do you think that oxaloacetate has got the phosphate group attached?
No, ma'am.
So, but we have used ATP over here. So, can you tell me the reason? वहाँ पे क्या हुआ exactly?
No idea, ma'am.
हाँ? Can you see it over here? क्या दिख रहा है वहाँ पे?
Ma'am, phosphate ion is released.
ATP is breakdown into ADP and phosphate, ma'am.
Yes, but it is not being utilized in the like transfer. It is not being transferred to the oxaloacetate, right? But the ATP has been used. So what does it mean? It means the conversion of pyruvate to oxaloacetate is high energy consuming step. Okay, even though it is not being transferred, but to carry out this, it was required a high energy. Then again, what happens over here? So oxaloacetate to phosphoenol pyruvate, again GTP is there. So GTP is also energy like ATP. Again, it was used. Right. Now, phosphoenol pyruvate has got your phosphate group. Okay, phospho, right? So that group is present now. So to convert from pyruvate to phosphoenol pyruvate, जो एक step में होना चाहिए था, 2 step में लगा और double the amount of energy was required. यहाँ पे क्या हुआ था? Energy release हुई थी. यहाँ पे energy double use हो रही है. So now from this you will understand that the gluconeogenesis path is going to be very costly, है ना? तो कोई भी चीज synthesize करना है तो इसका मतलब आपको पैसा तो लगाना ही पड़ेगा. तो यहाँ पे क्या आपका energy लग रहा है. Okay. Now then these steps are same. Phosphoenol pyruvate 2,2-phosphoglycerate 3-phosphoglycerate same since you can see that it is reversible. Okay. Now over here then 3-phosphoglycerate to 1,3-bisphosphoglycerate. Again ATP is being used. Now coming to this glyceraldehyde 3-phosphodehydrogenase, you will get your dihydroxyacetone phosphate, glyceraldehyde 3-phosphate, same aldolase. Now coming to fructose 1,6-bisphosphate. So PFK. Abhi kya hai? Here we have got the 2-phosphate group is there. I need to go back to the 1-phosphate which is present on the fructose at the 6th carbon, right? So of course here it was kinase which added the phosphate group. I'm going to use now phosphatase which is going to break the or remove the 1-phosphate group. Okay, so fructose bisphosphatase. So 1 phosphate group has been removed. So you will get fructose 6-phosphate. Then again phosphoglucose isomerase will give you glucose 6-phosphate. And then finally phosphatase to remove the phosphate group, you get the glucose. So always remember kinase means it is going to add phosphate group. and then you have got the phosphatase to remove the phosphate. ठीक है? So guys, you think that ये आपको gluconeogenesis का path can you remember?
Yes, ma'am.
हाँ, अभी याद हो गया है? Because once we proceed, because you need to remember then this. कि कब कौन सा क्या आया था? Because we are going to discuss gluconeogenesis in detail now, right? So anyways, I'll be sharing the PPT with you today itself so that अगर कोई doubt है तो you can just go through once and ask me later. Okay? So I'll be proceeding now. Okay. So I mean the pathway is clear to you, है ना? यहाँ पे कुछ doubt नहीं है आपको, right?
Yes, ma'am.
Yes, ma'am.
Okay. Okay, now conversion of pyruvate to PEP. So this is the first step in gluconeogenesis, right? So, so the first step in gluconeogenesis is the conversion of pyruvate to phosphoenol pyruvate or pyruvic acid. In order to convert this, there are several steps and several enzymes. So पहला कौन सा हो गया आपका pyruvate carboxylase, phosphoenol pyruvate carboxy kinase, and malate dehydrogenase. They are the three enzymes responsible for this conversion. So pyruvate carboxylase is found on the mitochondria, converts pyruvate into oxaloacetate. So guys, ये आपको थोड़ा सा ध्यान देना होगा यहाँ पे. So pyruvate carboxylase, ठीक है? Carboxylase मतलब यहाँ पे एक carbohydrate यानी कि CO2 is being added. So pyruvate carboxylase is found in the mitochondria. और आपका pyruvate कहाँ पे है? Pyruvate कहाँ पे होगा? Glycolysis happens in the cytoplasm. So your pyruvate is present in the cytoplasm, है ना? Okay. So what will be the first step then?
Yes, ma'am.
Pyruvate को आपको mitochondria में ले जाना होगा so that it can convert pyruvate into oxaloacetate. ठीक है? तब आपका जो pyruvate है to oxaloacetate में convert होगा. Where in the mitochondria, pyruvate is present in the cytoplasm. So first step will be to take the pyruvate from the cytoplasm to the mitochondria. Now, because oxaloacetate cannot pass through the mitochondrial membrane, it needs to be first converted into malate by the malate dehydrogenase. Then malate will cross the mitochondria membrane into the cytoplasm, where it is then again converted back to oxaloacetate with another malate dehydrogenase. अब oxaloacetate जो है वो phosphoenol pyruvate में convert होगा with the help of the Pep Carboxy Kinase. ठीक है? And then we have got the reverse of the glycolysis. Guys, ये आपको बहुत अच्छे से याद रखना होगा, ठीक है? So what is happening first? So your pyruvate is present in the cytoplasm. Okay? So this will be taken into the mitochondria. It will be converted to oxaloacetate by which enzyme? Pyruvate carboxylase. ठीक है? Now this will be converted to the malate by malate dehydrogenase. Why? Because oxaloacetate. Okay, so let me just repeat once again. So pyruvate, which is there in the cytoplasm, will be transferred to the mitochondria. Okay, now in the mitochondria, with the help of your pyruvate carboxylase, it will be converted to oxaloacetate. Now oxaloacetate cannot cross the mitochondria, so this will be converted to malate by malate dehydrogenase. Now this will cross the mitochondria, will come into the cytoplasm. Now in the cytoplasm, again with the help of the malate dehydrogenase, this will be converted back to oxaloacetate. Now with the help of your Pep Carboxy Kinase, it will be converted to your Pep, phosphoenol pyruvate. So now finally the first step in the gluconeogenesis has been done. What is that? Conversion of pyruvate to phosphoenol pyruvate. Okay, so I'm clear here till here.
Yes, ma'am.
Okay. Now about the pyruvate carboxylase. So it requires biotin. Biotin is a cofactor that is covalently attached to a lysine residue. It is a carrier of activated carbon dioxide, just as acyl-CoA carries activated acyl group. So that's why carboxylase, it is going to add the carbon group, right? So carbon dioxide group will be added. Okay, ये भी important है. Okay, for 2 marks question they might ask. Now, this is the entire paper which we were discussing about the conversion of pyruvate to Pep. So here, let's start. Okay. from where to start? Here you have got. So either if it is alanine or lactate, okay, later we'll discuss how alanine and lactate is getting converted to pyruvate. So we'll discuss that later. Okay, so once you get the pyruvate, okay, so that will be taken inside. The pyruvate will be converted into oxaloacetate to the malate. Okay, this is your mitochondria. Oxaloacetate to the malate. Now, from the malate to a malate will be taken out in the cytoplasm, again back to oxaloacetate, and then it is converted to your PEP, phosphoenol pyruvate, with the help of the carboxy kinase. And what you get is over here is the glucose synthesis. Okay, itna part clear hai? Till now what we have discussed is only this much. Okay.
Yes, ma'am.
So, and we'll discuss about this when we talk about the like from the fat and all, from glycogen, how can you get the glucose and all. So we'll discuss this later. This is also important to understand, okay, which is activated by, inhibited by, because all these enzymes, there are reasons like how they are getting activated or not. For example, let's talk about the glucagon. So the glucagon is your hormone, right? So it is going to activate your pyruvate kinase. So if it is activating pyruvate kinase, what does it mean? No, sorry, it is inactivating. It is going to inactivate. Anyways, we'll talk about this in the further upcoming slides. Here in the same figure, you might get confused. Okay. Now, regulation of pyruvate carboxylase. So, carboxylase is essentially activated by acetyl-CoA. Okay. So, when the concentration of ATP is low or the concentration of acetyl-CoA is low, then pyruvate is directed into the citric acid cycle to promote the synthesis of ATP. Now, high concentration of ATP and acetyl-CoA are signals that the cell energy level is high and metabolites are converted into glucose. If the energy status of the cell is low, then the concentration of ATP and acetyl-CoA are low, and thus pyruvate is directed towards the TCA cycle for the production of more ATP. Guys, is clear hai? So what does it mean from this? What do you understand? Pyruvate carboxylase. तो अगर एक्टिवेट करना है तो कौन कौन होना चाहिए? एसिटाइल-CoA एंड एटीपी. सो इनका कॉन्सेंट्रेशन लो होना चाहिए. ठीक है. कोई डाउट है?
No, ma'am.
हाँ, सिंपल है ना. क्योंकि अगर आपके पास एटीपी ज्यादा है, एसिटाइल-CoA ज्यादा है, तो क्या करना है? हमें इतना सारा एटीपी साइटोप्लाज्म में रख के, we don't want to make our cell as a bomb, right? Bomb of energy. So we are going to convert back into the metabolite. हम उसको ग्लूकोज में कन्वर्ट कर देंगे. जो भी हमारे इधर उधर जो मेटाबोलाइट्स रहेंगे, हम उसको ग्लूकोज में कन्वर्ट कर देंगे. और अगर हमारे पास स्टिल बहुत सारी एनर्जी है, तो ग्लूकोज को हम ग्लाइकोजन में रख के स्टोर कर लेंगे. So that whenever the time comes, like when there is no food available, so that time we need all this thing for the future thing, right? Okay. Now, Pep Carboxy Kinase, it simultaneously decarboxylates and phosphorylates oxaloacetate and forms the PEP. The decarboxy reaction is very exergonic and helps to derive the otherwise highly endergonic reaction of Pep synthesis. See, this is what decarboxylation reaction में इतनी energy है that it is sufficient to carry out this endergonic reaction of Pep synthesis. Because हमने phosphate को वहाँ पे add किया, right? Endergonic means it needs energy. Exergonic releases energy. So this energy releasing step is sufficient enough to derive this endergonic reaction of Pep synthesis. Now, compartmentalization of pyruvate carboxylase in Pep Carboxy Kinase. Now, why we are talking about this? Because each tissues and cells, they have got different location where these carboxylase and carboxy kinase are present. Carboxylase का क्या काम था? To convert pyruvate into, yes, क्या काम है? Pyruvate into
Oxaloacetic acid.
Yes, and carboxy kinase का काम है to convert oxaloacetate into your PEP, right? So now just listen carefully. Try to imagine the entire mitochondria. क्योंकि हम matrix, फिर उसका transmembrane, intertransmembrane region, then cytoplasm, all this we are going to discuss. So if you remember, if you can understand where these enzymes are located, then it becomes easy. Although in the figure which we just now saw, उसमें दिया हुआ है, again it's the same thing, just with the theory. So if you can imagine and remember, then the questions which comes in the exams will be easy. Okay. सो कंपार्टमेंटलाइजेशन मतलब कहाँ कहाँ हमको डिफरेंटली लोकेशंस में रखा गया है, ठीक है। So the pyruvate carboxylase is found in the matrix of the mitochondria. So इससे हमें समझ में आता है कि yes, pyruvate को हमें साइटोप्लाज्म से माइटोकॉन्ड्रिया में ले जाना है, and then only we can convert pyruvate into oxaloacetate. So it means the pyruvate carboxylase आपका matrix में. Okay. The next enzyme involved in the gluconeogenesis is Pep Carboxy Kinase. It is found both in the mitochondria or in the cytosol, depending on the organism and the tissues. ठीक है. Now in human livers, Pep Carboxy Kinase is found in both the mitochondria and cytosol. तो यहाँ तो हमें कोई दिक्कत नहीं होनी चाहिए, राइट? द फॉर्मेशन ऑफ ऑक्सैलोएसिटेट एंड पाइरुवेट का कहाँ पे? ह्यूमन लिवर्स में. So pyruvate is transported into the mitochondria where it is converted into acetyl-CoA by pyruvate dehydrogenase and enters the citric acid cycle. ये हो गया आपका normal, okay, glycolysis. और it is converted into oxaloacetate by pyruvate carboxylase and used for gluconeogenesis. इतना clear है? This paragraph is clear, guys, from here to here. Okay.
Yes, ma'am.
Now in tissues where Pep Carboxy Kinase is found only in the mitochondria, तो वहां पे क्या होगा? Oxaloacetate needs to be converted into Pep in the mitochondria only. तो कैसे? वहीं पर आप मैलेट बनाओगे, वहीं पे आप कन्वर्ट करोगे, ठीक है. अगर आपका पेप कार्बोक्सी काइनेज जो है साइटोसॉल में है तो आप क्या करोगे? ऑक्सैलोएसिटेट को मैलेट में कन्वर्ट करोगे, मैलेट को साइटोप्लाज्म में लाओगे, उसके बाद अगेन ऑक्सैलोएसिटेट में कन्वर्ट करोगे एंड देन यू आर गोइंग टू कन्वर्ट इनटू पेप, ठीक है. And that's how your gluconeogenesis will be done. Clear, guys?
Yes, ma'am.
Now, because the inner mitochondrial membrane is impermeable to oxaloacetate, cells which lack mitochondrial Pep Carboxy Kinase transfer oxaloacetate into the cytoplasm by using the malate shuttle. तो यहाँ पे हम malate shuttle के बारे में थोड़ा सा डिस्कस करेंगे. So what is happening in the malate shuttle? So in this process, oxaloacetate is converted into malate by the mitochondrial malate dehydrogenase. And after the transfer, the reverse reaction is catalyzed by the cytoplasmic malate dehydrogenase. So guys, इसे ये मत समझना that mitochondrial malate dehydrogenase will also come into the cytoplasm. No, we already have malate dehydrogenase in the cytoplasm as well. Okay. So the malate shuttle allows the gluconeogenesis to continue because it provides NADH required for reaction catalyzed by glyceraldehyde 3-phosphate dehydrogenase. Guys, जहाँ जहाँ dehydrogenase है वहाँ आपका NADH लगेगा, है ना? मैंने हमेशा बोला है. Now, after your phosphoenol pyruvate is formed by the Pep Carboxy Kinase, it is hydrated by enolase to form 2-phosphoglycerate. फिर क्या होगा? 2-phospho आपका 3-phosphoglycerate बनेगा, and then you are going to get 1,3-phosphoglycerate. फिर वो glyceraldehyde 3-phosphate बनेगा, and then the reverse of glycolysis will happen. ठीक है? So all these enzymes, they are nothing but the reverse of glycolysis is happening. So guys, I guess आपको इतना clear हो गया है till we get fructose 1,6-bisphosphate. वो सारा का सारा आपका glycolysis and gluconeogenesis है. तो अभी हमारा इतना clear हो गया है ना?
Yes, ma'am.
Now, conversion of fructose 1,6-bisphosphate to fructose 6-phosphate. So as I said, ये आपका PFK1 ने किया था, phosphofructokinase ने किया था, right? So we need to reverse it. ये आपका irreversible reaction है. So that's why you need another enzyme. So the phosphorylation of fructose 6-phosphate by phosphofructokinase is the first commuted step of glycolysis, and it is irreversible. So for gluconeogenesis, fructose 1,6-phosphate is hydrolyzed by the enzyme fructose 1,6-bisphosphatase. So hydrolyze हुआ है. So it means water molecule is being added. ठीक है? Okay. The standard change in free energy for this reaction is given. So what does it mean? अगर ये negative है, so it means it can be carried out then. So the irreversible PFK1 catalyzed reaction glycolysis is bypassed by the fructose 1,6-bisphosphatase enzyme, bisphosphatase. So this enzyme is present in the cytoplasm of liver and kidney, and it is exergonic. Exergonic मतलब energy releasing reaction. So minus जब भी होते हैं, it means it is exergonic. Energy आपकी release होती है. Plus है तो मतलब energy आपकी use हुई है. So इसके लिए अलग से हमें ATP use करने की जरूरत नहीं है, because breaking the phosphate bond itself releases a lot of amount of energy. Okay. So finally, from your fructose 1,6 bisphosphate, you are getting your fructose 6 phosphate. अभी क्या है? इसके बाद isomerase. So fructose 6 phosphate will give you glucose 6 phosphate. Okay. Before that, so this exergonic reaction is also irreversible under cellular condition. ATP is not regenerated and inorganic phosphate is released in the cytoplasm. हाँ, क्योंकि phosphate group is removed from the fructose 1,6 bisphosphate, right? So one phosphate group is released, that is inorganic phosphate is released. Now it is a allosteric enzyme. So like other allosteric enzyme, its activity is stimulated by citrate and inhibited by AMP and fructose 2,6 bisphosphate. Okay, so why the activity is stimulated by citrate? अभी हमने mention किया था, if we have got many metabolites, they can be used for the synthesis of glucose, right? Inhibited by AMP. क्यों? अगर ज्यादा amount of AMP है, if suppose you have got high concentration of AMP, so should I start making glucose or should I start undergoing glycolysis? Yes, guys.
Ma'am, we will undergo glycolysis.
Yes, because we need ATP, right? So अगर आपके पास AMP है तो मतलब मुझे ATP चाहिए, तो मैं तब ग्लूकोज नहीं बनाऊंगी, तब मैं ग्लूकोज यूज करूंगी because I need energy. And same way with fructose 2,6 bisphosphate. So from here you will understand कि अब fructose 1,6 bisphosphate को कौन एक्टिवेट करेगा? सिट्रेट एक्टिवेट करेगा और AMP उसको इनहिबिट करेगा. ठीक है? Now so now about the formation of glucose from the glucose 6 phosphate, right? So this is our last step. So the glucose 6 phosphatase enzyme, which is found in the ER of liver and kidney, catalyzes the irreversible hydrolysis of glucose 6 phosphate to glucose and PI. So subsequently, this glucose is then released into the blood and taken by the cells. तो भी सेल के अंदर भी पहले हमने देखा माइटोकॉन्ड्रिया यूज होता है, अभी क्या है? इन दी एंडोप्लाज्मिक लिवर इट इज हैपनिंग. ठीक है, एंडोप्लाज्मिक रेटिकुलम ऑफ लिवर. Okay, so glucose 6 phosphate is a phosphatase. It is going to remove your phosphate from the 6th carbon, and finally what you get is phosphate glucose in the finally you are going to get the glucose. अब जहां जहां आपको ग्लूकोज की जरूरत है, so your blood is going to take the glucose and to the final destination. जहां पे भी आपके cells and tissues को requirement है, वो वहां पे उसको ले जाएगा. ठीक है? Okay. So from this I can say that, I mean I can tell you that the gluconeogenesis is completed. Okay, so हमने glycolysis देख लिया, gluconeogenesis देख लिया, and if you remember in the previous class we also talked about like the connecting link between the gluconeogenesis and glycolysis is called dash. कुछ याद है क्या कहते हैं उसको?
Ma'am, Cori cycle.
Yes, very good, Cori cycle. Okay, so the energy balance finally. Gluconeogenesis is energy consuming and glycolysis आपका ATP generating है. ठीक है, तो अगर हम पूरा कैटलाइज करें, पूरा देखें कि क्या हो रहा है, तो glycolysis में क्या होगा? ग्लूकोज and finally you get 2 pyruvate, so NAD plus है ADP plus 2 PI. Finally what you get is this. Gluconeogenesis में 2 pyruvate NADH ATP GTP water molecule सब यूज हुआ है. At the end what you get is glucose. ओवरऑल अगर हम देखेंगे तो क्या हुआ है? 2 ATP 2 GTP एक्स्ट्रा हमारा यूज हुआ है. ठीक है, बाकी सब आपका कैंसिल हो जाएगा. बट इफ आई डू वन राउंड ऑफ ग्लाइकोलाइसिस इन वन राउंड ऑफ ग्लूकोनियोजेनेसिस, सो व्हाट इज द नेट गेन? आई एम इन द लॉस ऑफ 2 ATP एंड 2 GTP मॉलिक्यूल. ठीक है, तो ग्लाइकोलाइसिस से जो 2 ATP मुझे मिलता था, अब क्या होगा? एक्स्ट्रा मेरे 2 ATP और 2 GTP और चले गए. सो इन एंड ऑल इट मींस ग्लूकोनियोजेनेसिस इज हाईली एनर्जी कंज्यूमिंग प्रोसेस. ठीक है, तो ऑफकोर्स सी बात है ना, बिकॉज कोई भी चीज बनाने में आपको एनर्जी पैसा तो लगता ही है. सो दिस इज व्हाट इज हैपनिंग. नाउ दिस इज आल्सो इंपॉर्टेंट गाइस, बहुत बार ये क्वेश्चन आ चुका है. सो पेशेंट्स विद वॉन गिल्ट डिसीज दे लैक ग्लूकोज 6 फॉस्फेटेज एक्टिविटी इन द लिवर. ग्लूकोज 6 फॉस्फेटेज यानी कि क्या? ग्लूकोज 6 फॉस्फेट से ग्लूकोज नहीं बन सकता. इट मींस द चाइल्ड और द पेशेंट कैन नॉट अंडरगो ग्लूकोनियोजेनेसिस. ठीक है, so this resulted in the accumulation of excess glucose in the form of glycogen. Upon fasting, the patients are not able to convert this glycogen into glucose. Thus, fasting, hypoglycemia, and lactic acidosis is two prominent symptoms of this disorder. क्यों? Because आप fasting कर रहे हो, जितना भी आपका glycogen है वो यूटिलाइज ही नहीं हो रहा है, राइट? और जिसकी वजह से क्या होगा? ग्लूकोज शरीर में नहीं मिल रहा है. बॉडी में नहीं है, it will result in the hypoglycemia. हमेशा the patient is going to have this problem of hypoglycemia, जिसकी वजह से सीजर्स होगा, कोमा में जा सकता है, there won't be any intellectual disability. क्यों? Because glucose is required for brain activity, and अगर आप ग्लूकोज ही नहीं दे पा रहे, so the brain will have its own disorders, right? Now when the glycogen results in the accumulation, यानी कि क्या ग्लाइकोजन आपका यूज नहीं हो रहा है, so it will result in the kidney enlargement. Hepatomegaly, it means the liver आपका जो है enlarge हो रहा है. So liver disease, kidney disease, everything this patient is going to face. ठीक है? So they are called bone to Gierke disease, जिसने एक person discover किया. So please remember this also. Okay. So here I can say that the gluconeogenesis has been completed, and there are few more topics like how, like what are the substrates which can be used for the gluconeogenesis that we'll be discussing tomorrow. Okay. Yeah. So guys, any doubt in today's lecture? I'll be stopping here now.
No, ma'am.